Good afternoon, everybody. Welcome to the second lecture. So we're going to be talking about RNA. RNA differs from DNA in that instead of using deoxyribose, which is the carbohydrate of DNA, we use ribose. And in place of thymine, which is found in DNA, we have uracil. Also, RNA is typically much shorter than a DNA strand, and RNA is always single-stranded unless we're looking at viral RNA structures. So RNA is synthesized from DNA, and there are 3 different types of RNA. So we have messenger RNA, which carries a message of DNA. So it's going to carry the same information that the DNA has, but in an RNA format, which our cells can read. There's ribosomal RNA. You see these are the ribosomes, which carry out protein synthesis. And then we have the transfer RNA, which proteins are composed of amino acids that are bonded together. Transfer RNA carries the amino acid. Carries that appropriate amino acid to the ribosome, helping to form that long polypeptide of amino acids, which will fold into a functional protein. So in bacteria, the DNA that is found is typically found in a circular structure. In general, bacteria only have 1 chromosome, and it is a double-stranded circular chromosome. And so this is, you can see in the figure what the chromosome looks like. In replication, we're going to start replication and at a region called the origin of replication. Replication occurs in both directions because replication always is, or DNA is always produced in a 5' to 3' direction. Apologies, I will stop my dogs from barking or making noise, and I'll be right back. My dogs like to play. So if you're wondering, that was Frank and Dudley. They love playing. Yep. Hello. Go to the next slide. There we go. So there's a lot of different enzymes that are used in DNA replication. Replication seems like a very simple process, but it actually takes a coordinated effort on multiple different proteins. And so this is just a list of some of those structures and some definitions of structures that we're going to produce. So replication. Before we start replicating, the 2 strands of DNA have to unwind and unzip. So DNA is bound together. The 2 complement strands are bound together by hydrogen bonding. So when we break those hydrogen bonds, that is called denaturing the bonds, okay? So the term denature, which you've learned probably in your chemistry and gen bio classes, is breaking proteins down. But when you denature, or when you break the hydrogen bonds in DNA, that is also called denaturing. Okay. And so the enzyme helicase is the one that unwinds and helps with the unzipping of the hydrogen bonds. So it opens up the DNA so that we can then copy it. Once we have the DNA starting to open and starting to unwind, topoisomerase is an enzyme that breaks DNA molecules apart so that they can separate easier. And then we have primase, which is an RNA primer that produces a small structure that binds to the DNA. DNA polymerase has to bind to a double-stranded structure. So it can't bind directly to the single strand of the DNA. So we have that primer that allows DNA polymerase then to bind. And then DNA polymerase binds and it starts producing complements to the parent strand. So here we have helicase right here. Unwinding and unzipping the DNA. And I want you to note that the red strand is the 5' end and the blue strand is the 3' end because that's important. We have primase laying down RNA primers, and you can see one right there. And then we have DNA polymerase. You can see one here. You can also see one right here, producing a new complementary strand. Notice that on one of the strands of one of the parent strands, the red one up here, we have a continuous production of a new strand, because down here we have that 3' end, because remember up here it was 5' to 3'. DNA reads in a 3 to 5' direction, and it lays down new strands in a 5 to 3 direction. We always produce DNA in a 5 to 3' direction. And then notice on the other side we have that 3' end over here. Oh, sorry. We have that 3' end over here, and so that means down here we have a 5' end, right? Well, DNA only reads in a 3 to 5 direction, which means it has to read closer to that helicase. So as helicase is unwinding, it lays the RNA primase lays down a primer, and DNA polymerase can produce a section of the DNA. But then as helicase opens more, another primer is laid down and DNA polymerase produces another section. And then another primer is laid down and DNA polymerase produces another section. And so this strand up here is called our leading DNA replication strand. And this strand down here is called the lagging DNA replication strand, okay? And I'm going to stop here because this has been a 9-minute video, and we'll get back into replication in the next video, okay? Bye.